Thank you.
So Bikash, are you recording? Greetings to all and a warm welcome to the ZOOMinar and the molecular basis of proteinopathies. Today's speaker is Professor Dennis Selkoe from Harvard Medical School. We also would like to welcome guests to the panel, Dr. Henry Paulson from the University of Michigan, Dr. Rakesh Kayat from the University of Texas Medical Branch, Galveston, and soon joining Professor Yehud Gazit from Tel Aviv University. Today's speaker, Dr. Selkoe, is quite well known to this audience and doesn't need any introduction, but I will do the formality. Dr. Selkoe is a graduate of Columbia University and the University of Virginia School of Medicine. He was trained at NIH and at Harvard Medical School. Dr. Selkoe's many exciting discoveries on the cause and mechanisms of Alzheimer's disease include the formulation of the amyloid hypothesis, the production of amyloid beta by normal cells throughout life, the mutations in amyloid precursor protein, and later gamma secretase cause Alzheimer's disease by increasing A beta production. His team has also investigated alpha-synuclein, the key misfolded protein of Parkinson's disease. It's obvious that he has received many honors and awards that includes H. Heineken Prize for Medicine, the Mathilde Solovey Award in the Neuroscience from NIH, the Potemkin Prize from the American Academy of Neurology, the Pioneer and Lifetime Achievement Award from Alzheimer's Association. He is also a member of the National Academy of Medicine. He is now the founding director of Proteinopathies Biosciences. With that brief introduction, I'm pleased to welcome Dr. Selkoe to present his lecture. Dr. Selkoe.
Great. Thank you very much, Rams, and I hope that everyone can hear me.
Yep.
Speak up if you can't for any reason. So I'm very happy to be with you virtually. I prefer to be in Ann Arbor. I'm a Midwesterner, and I actually have a summer house in Glen Arbor and on Glen Lake near the Sleeping Bear Sand Dunes. So I often come to Michigan. For those of you who happen to be in Michigan, I know you're all over the place. And so Michigan has always been dear to my heart. But I'm happy to join you today to speak about, I think, the key topic for this series, which is the molecular mechanisms of protein misfolding disorder. And today I'm going to talk about our work on Alzheimer's and Parkinson's and how we believe that this type of work, not just in our lab, but many labs, will lead to disease-modifying therapies. So the theme of my overall approach is very much going to pathophysiologically relevant in vitro and in vivo systems. That is, less focus on synthetic assemblies and more on whatever we can harvest naturally from the brain tissue of patients with these diseases. So that's the overall theme of going to pathophysiologically relevant endogenous type species. I'll mention that I have only one disclosure to make, and that is I'm a director and consultant to Proteinopathies Biosciences, which Rams mentioned, but that has nothing to do with this work, and none of this work is done really other than with my lab and its collaborators. So I'm going to begin by telling you quickly something that you're probably familiar with already, and that is the notion that, like other amyloid diseases, the Alzheimer amyloid process, which I'll speak of first, relates to the aggregation of the amyloid beta peptide that happens with age in all primates. And it happens actually in dogs and cats. So if you have dogs and cats at home that are north of their late teens or mid-teens, they're likely to have the same process that we're describing here. In a canonical cell, cartooned in yellow here, the cell wall may have the amyloid precursor protein, in red and black. And many of you are aware that there
Dennis?
Yes.
Can you hear me?
Yes,
I'm not seeing your slides. I don't know if you have slides up yet. Are you trying to share your slides?
I do have slides up, yes.
Okay. Well, I don't think any of us are seeing them.
Okay.
Ron, are you seeing them?
This is the first slide of his. Did you forward the slide or?
No, so I'm doing my own screen share.
Yep, it's fine. Yeah, everything's working good.
Well, none of us can see the screen.
No, Oh, really? I'm able to see the screen. Sam, are you?
No, I cannot see the screen.
Dr. Selkoe, maybe stop sharing and share again.
Okay, sure. All right. All right, can you see my face?
Yep.
Yep.
Okay, so I'm going to share again. Okay,
Now we're good.
Oh, that's good.
All right, good. So it just needed to be started again. It was started about 15 minutes before the lecture. So let me put me in slideshow. So what I'd shown you so far was this image. Can you see the blue and green image?
Yep.
Yes.
Okay, good. And I had just declared the fact that I'm a director and consultant to Proteinopathies, which is irrelevant here. And now I'm going to this canonical cell, this yellow image of a round cell wall with the amyloid precursor protein embedded within it. And the A-beta region is right within, in part, the transmembrane region. And there's an orderly cleavage process that we discovered in 1992 by beta secretase cleaving and releasing a large ectodomain fragment in the extracellular fluid, or if this is in the lumen of a vesicle, it would be the interior of the vesicle, the lumen. And then a second very curious cleavage occurs in the membrane called gamma secretase. And these were the activities that were nicknamed alpha, beta, and gamma secretase without knowledge of what the cognate enzymes or proteases were. This second cleavage is in the membrane and it releases the amyloid beta peptide from APP. And as the title of this slide shows, this is entirely normal and occurs throughout life, and essentially almost all animals express APP. It's ubiquitously expressed, and certainly all primates develop A-beta accumulation with age. And the A-beta monomer itself, we believe, is not toxic. We know little about its precise molecular function, but there's some evidence that it can act as a growth factor or neurotrophic factor, ironically. That's when it's a monomer, but when A-beta dimerizes, which can happen inside the lumen of a vesicle, inside a cell, or in the extracellular space, A-beta dimers form. And this is something that we believe does not happen in young animals or young humans, but happens more with time and with age. And age, of course, is the #1 factor for the appearance of Alzheimer's disease. Dimers and trimers and tetramers and larger oligomers are collectively formed by A-beta under pathological conditions. So the A-beta monomer is normal. The A-beta oligomers are viewed as abnormal or pathological. And those oligomers can, under some circumstances, polymerize into protofibrils and fibrils. And the protofibrils and fibrils form amyloid plaques in the patient's brain. So this is the pathological aspect of A-beta production. And that leads to the classical pathology that you see here. These are two spherical deposits of amyloid beta protein filaments or fibrils, which I'm pointing out here with my cursor. And these two spherical deposits are surrounded by dystrophic axons and dendrites that you see as squiggly dark lines. And adjacent are neurofibrillary tangles. And this is the modified Bilshowski stain that indeed a colleague of Bilshowski, Alois Alzheimer, working in the Kreplin laboratory in Munich, first used to describe the first case of the disease that now bears his name. So amyloid plaques with neuritic dystrophy around them or within them and neurofibrillary tangles are the classic pathology since 1906, when Alzheimer called attention to it, and it's still used by neuropathologists today. Now, let me delve right away into the molecular mechanisms of Alzheimer's as I see them. A key finding is the notion that presenilin-1, one of the genes discovered in 1995 as a site of mutations associated with familial Alzheimer's, Presenilin 1 is actually an intramembrane aspartyl protease. And here I show the single letter amino acid code of all 467 amino acids. And we've expanded TM6 and 7 of this polytopic presenilin protein that was first brought to attention by Peter St. George Hislop at the University of Toronto when he discovered that mutations in this gene and its protein can cause aggressive early onset familial Alzheimer's. That was in 1995, and in 1999, Michael Wolff, Wei-Ming Shah, and I hypothesized and then demonstrated that 2 and only 2 aspartates, the Ds you see here in the intramembrane regions of TM6 and 7, were a novel intramembrane aspartyl protease. This was the first example of an intramembrane aspartyl protease and was combined with the evidence from Brown and Goldstein of the Site2 protease for SREBP processing in cholesterol homeostasis, and they coined the term regulated intramembrane proteolysis. So the way that this works is that these 2 aspartates that are charged residues intramembranous can coordinate with a substrate, a helically wound substrate that crosses the membrane, a transmembrane protein, and effect an aspartyl protease cleavage. And this was very controversial when we proposed this based on the structure here, but it subsequently was shown in biochemical experiments, and I won't go through those. This is some considerable time ago. This cartoon demonstrates what we meant, and that is in yellow and orange is the presenilin molecule that winds its way in and out of a bilayer 9 times. And the 2 aspartates in TM6 and 7, we hypothesized and then demonstrated were the active site of an unusual intramembrane aspartyl protease that cleaved helically wound substrates. And in this case, my cartoon shows the APP molecule lying in a groove-like structure that has been subsequently shown by cryo-electron microscopy reconstruction to be indeed present in presenilin. And the beta secretase enzyme would have cut already in this cartoon here at the base of the helix and removed the large ectodomain. And the remaining C-terminal portion of APP lies in this groove and undergoes a cleavage by the 2 aspartates at this position, releasing the purple part in this cartoon, which is the A-beta peptide. So this is a cartoon that demonstrates what we believe to be the mechanism of gamma secretase, AKA presenilin. This work then in 1999 and subsequent work from a number of laboratories supported the notion that presenilin causes Alzheimer's disease because it is the actual protease that cleaves APP to release A-beta. And mutations in this protease can heighten the amount of A-beta released and or can lengthen the A-beta peptide that's released. Rather than the more common 40 residue, there would be more 42 amino acid A-beta or 43, or even longer potentially. This provided a major linchpin for the amyloid hypothesis that 2 or 3 of us had put forward in the early 2000 period. And that is that all of the mutations that cause dominantly inherited Alzheimer's disease occur either in the substrate, APP, or the protease, presenilin, AKA gamma secretase, that affects the production of A-beta. So A-beta production is a direct consequence of these dominant missense mutations in the substrate or the protease. So what I've discussed very briefly is one aspect of the life cycle of A-beta, namely the production of A-beta from beta secretase cleavage of APP followed by presenilin gamma secretase cleavage within the membrane. And that happens normally. And if one degrades or variant seems to do, it does not allow proper clearance of A-beta from the extracellular fluid. the A beta can aggregate. And I'm going to speak about that now because that's a very important aspect of Alzheimer's. Indeed, there's a special role for bioactive A beta oligomers, dimers and larger, whereas the A beta monomer, as I've mentioned, is a normal physiological product. So with time, one accumulates A beta monomers and dimers in the case of the disease called Alzheimer's disease. And aged normals, here control 1 and control 2, of course, produce some A beta. One can hardly see it at this level of Western blot exposure, but far less monomer accumulates in the brain. So these are simple saline extracts of Alzheimer's tissues, 84, 85, 86, 87, patients whom we've followed during life or others have in our center. And we made simple saline extracts and ran them out and electrophoresed them and Western blotted them for A beta. You can see the monomers and dimers of A beta. If you take a fraction like the one on the left, AD4, of a physiological buffer saline extract, you can show that the extract inhibits LTP, shown in red. This, of course, is the phenomenon of long-term potentiation, which has been known by neurobiologists for decades now to be a electrophysiological correlate of learning and memory. And the control brain extract in blue does not impair LTP, whereas the vehicle also supports long-term synaptic potentiation with this electrical stimulation. Now, if you first remove the A beta material and only the A beta material by a selective immunoprecipitation, shown in lane 2 of this small Western blot, now the Alzheimer extract no longer impairs LTP. And you get the normal synaptic plasticity or potentiation in these wild-type mouse hippocampal slices. So this is to demonstrate that material from human brain can impair long-term potentiation in a wild-type hippocampal slice. And we also showed a little later that one can induce with a certain stimulus frequency a long-term synaptic depression, shown in red here, when one applies an Alzheimer brain extract, again, such as the one in lane 1 of the Western blot on the upper left. And that is not seen when one applies a control tris-buffered saline extract of Alzheimer brain. So these experiments suggest that something in Alzheimer brain extracts that is removed by A beta antibodies by immunodepletion can impair LTP and enhance long-term synaptic depression, which you see here in red. Now, we've done considerable studies, and so have a number of other labs, to lead us to images like this, cartoons that attempt to explain what's happening under the normal circumstances on the left in terms of presynaptic glutamate release in the glutamatergic synapse and the various receptors in the postsynaptic membrane, and that under some circumstances, electrical stimulation can induce long-term depression, but that happens much more readily in the presence of A beta. So on the right is the situation we imagine happens to synapses in the Alzheimer brain, where A beta dimers, trimers, tetramers, dodecamers, etc., are floating in the extracellular fluid of the brain and interfere with synaptic transmission. And in terms of the mechanism by which this alters synaptic transmission and induces enhanced long-term synaptic depression, our best evidence suggests that A beta oligomers are interfering with glutamate reuptake. So the glutamate transport here in green transporter is affected by binding of the A beta dimers. And so we view this as a excess extracellular glutamate that leads to extrasynaptic spillover, and NR2B receptors in particular, we have published, will be affected by the excess of A beta. And indeed, I should point out that there was beautiful work done in the laboratory of Arthur Konert in Munich a couple of years ago, in which he used A beta oligomers from human brain that my colleague Dominic Walsh prepared at the time and shipped to Munich to show that their injection into the ventricle, ICV injection, would impair glutamate reuptake. And that was a similar mechanism that he saw in vivo, as we had described in cells and in cultured cells and in slices. So although there are multiple mechanisms by which excess A-beta accumulation in the brain can lead to synaptic abnormality, one of those, and I think a prominent one, is interference with synaptic, postsynaptic glutamate reuptake, and also perhaps by astrocytes. Now, that's the electrophysiology that we've studied. What about actual behavior, function? In this set of experiments, we placed a cannula that you don't see here into these healthy adult Wistar rats, a cannula into the ventricle, cemented in place. The animal was allowed to recover from the surgery. That's shown here in this timeline. And 10 days later, we injected into the ventricle of the rat soluble extracts from our patients' brains. So this is sort of a Koch's postulates, if you will, sort of transmitting the material that we think is harmful to an experimental animal and learning what function it has. And we know that a rat will prefer to go into an open, dark space. And in this particular cage or fear avoidance chamber, it can get a single foot shock from the metal cage floor, and it'll scurry back into the more well-lit, dark, crowded chamber. And the animal will remember this. If you put it back into this apparatus 4 hours, 24 hours, 48 hours later, there will be variable degree of recollection that it would not be wise to go through the open hatch into the middle, the dark open chamber that it would otherwise prefer. When you inject oligomers of A-beta from patients' brains into the ventricle and wait 4 to 24 hours, the animal placed back in does not remember that it's an adverse environment on the left, and it scampers over here. And so we can time that, and did time it, and published in 2008 that soluble oligomers from human cortex will interfere with the performance of a learned behavior, namely avoiding this foot shock. So there's a behavioral or functional deficit in memory of a task in a healthy adult rat. And then we did experiments to connect these effects of A-beta oligomers on synaptic efficacy with the other major lesion that Alzheimer's described more than 100 years ago, and that's the tangles of tau. And the tau neuritic dystrophy was able to be modeled by using endogenous A-beta monomers and dimers, which we isolated by size exclusion chromatography from saline extracts of our patients' cortices. And here you see that in a control brain, there's no A-beta material in an age-matched control saline extract, and in the Alzheimer's saline extract, we can separate by SEC 8 kilodalton dimers and 4 kilodalton monomers of amyloid beta peptide. And there's a lot more complexity to this. I'm giving you just one example of a SEC profile. The material at the top of the gel is cross-reactive material that's not relevant to the A-beta process. So we can compare control, age-matched control human cortical extracts to AD saline extracts. And when we apply these to healthy neurons, we can do this with rat neurons, as in this image, or with human iPSC-derived neurons, one sees that only in the far right is there an abnormality of neuritic architecture. So by tau staining and tubulin staining, these neurons look very beautiful, and the application of the control brain extract, fraction 10 or 11, which would be the A-beta monomer equivalent, or the A-beta dimer equivalent fraction, doesn't impair things, because there isn't any A-beta in those fractions. A-beta monomers provide an excellent control from the Alzheimer's patient's saline extract, and they have no effect over several days. But the A-beta dimer-rich extracts, separated now, do have neuritic dystrophy as a consequence of that. And we can also see, as outlined in red here, that by Western blot, there is a heightening of the signal for an antibody such as AT8, which is an antibody that recognizes a phosphoepitope in the tau protein, the tau protein being the subunit of the tangles in Alzheimer's disease. And that increase in tau phosphorylation compared to total tau level on the Western, that can be eliminated if you first extract A beta oligomers from the extract by immunodepletion. Now you no longer get an elevation of phospho-tau. So as we say in the title, human A beta dimers and higher oligomers selectively increase tau phosphorylation at 80 relevant epitopes, the epitopes that one sees if one stains the brain with antibodies like this, shown in lane 4, and A beta immunodepletion prevents this in lane 6. So this ties together the A beta oligomer effects on synaptic efficacy with structural effects on neurites, neuritic dystrophy that we liken to the abnormal neurites around plaques. We've taken advantage of these observations to look at this from a therapeutic perspective. So over time, in an incucyte live cell imaging paradigm in the lab, you can see a color-coded image of neurites, of healthy human neurons, iPSC-derived neurons, and the progressive loss of neuritic length and intactness by applying these picomolar or low nanomolar amounts of soluble A beta oligomers over time. And this can be rescued in these images when you just float and certain anti-A beta antibody onto the slide. And this is the renowned antibody in the Alzheimer's field, Bananamab, that was in early clinical trials, was underdosed and did not have any benefit, but was sort of at the beginning of Alzheimer's immunotherapy. We've done this iteratively with Bananamab, as shown in this graph in the lower half, and with a number of others, BAN2401, Lecanemab, which has just gotten fast track approval from the FDA, that is the fast track status for possible accelerated approval in 2022. Adukanymab, which is much in the news as the first FDA approved immunotherapeutic. We've quantified the effect on neurite length in our cultures from applying these various antibodies. And you can see their rank order. And this is an irrelevant antibody, Avastin, used in macular degeneration, which does not do anything to the A beta toxicity. And these examples of the actual neuritic dystrophy measurements, the length of neurites are declining when you apply these A beta oligomer-rich brain extracts, but not in the presence of several of these putatively therapeutic clinical antibodies. Now, where is this type of analysis of the potential centrality of A beta oligomers to the Alzheimer's process, including the tangle formation? Where has this gone clinically? And we know that it's been a stormy and difficult route so far. This is the evidence published by Savigny et al. In Nature 2016. For the first phase 1, it's actually an advanced phase 1B or early phase 2 proof of concept study in 165 patients, which Biogen referred to as the PRIME trial of this human antibody, Adukanymab, directed against the N-terminal region of the amyloid beta peptide. And so like the neutralization experiments I just showed you in our cultures, in humans, there was a dose-dependent decrease over 1 year of treatment compared to placebo in the amount of A beta visualized by PET imaging in the cortex. So this is a steady dose-dependent decrease in A beta as a result of monthly treatments with, for example, the maximum dose of 10 milligrams per kilogram body weight per month for 12 months. So amyloid clearing visualized by PET amyloid scanning. This was measured here in a graph, again showing this dose-dependent. The higher dose is purple, the lower dose is blue, placebo is gray, and there is over 78 weeks a decrease in the amount of amyloid. And this decrease can be quite striking and was seen not only in this trial of Biogen's, the phase 3 EMERGE trial, but also in their other trial, ENGAGE. The problem was that EMERGE showed some evidence of clinical slowing of decline, but ENGAGE did not. And that has led to considerable concern and consternation about the fact that this was a split jury, although the FDA did decide to approve this based principally on the eMERGE data. They also took into account CSF tau levels. CSF phospho-tau levels, which are a marker of tangle formation, went down in a dose-dependent fashion in the eMERGE study. They also did in the ENGAGE study that wasn't clinically positive, but less so. So there was less lowering of tau, and it did not achieve significance in the other study. And the amyloid reduction was a little bit less also in that case. This is the clinical data on the trajectory of an activities of daily living scale, the ADCS ADL scale, which is how patients during the 78-week trial performed in various standard tests, preparing a simple cup of coffee or doing a certain task in everyday life. And there was about a 20 to 30%, in the case of ADLs, maybe a 40% less decline over 78 weeks. And this was the basis on which, in this one study by Biogen, eMERGE, FDA felt that there was evidence of clinical effect. Now, importantly, the FDA decision to do an accelerated approval of Adukanymab, even though the trial results in phase 3 were split, one with clinical benefit of modest degree, the other with no apparent clinical benefit despite amyloid clearing. The agency was influenced by this plot, which shows a number of clinical trials of the antibodies named here at the bottom of the graph. And I won't go over all of these, but this, I mentioned Bananamab earlier, which wasn't successful, Crenezumab and Solanezumab that don't really clear plaques well. And then going to the lower left in this graph, you're going towards a greater decrease in amyloid plaque burden on PET scan and a less decline, lower change in the mean difference between drug and placebo on the commonly used CDR sum of boxes, clinical dementia scale sum of boxes rating scale, or CDR scale. And so this trend among several different trials that you show here, Adukanymab and BAN2401 and Bananamab, impressed the FDA as evidence that plaque reduction was correlated to some extent with a small but significant benefit. The one outlier was the study I've mentioned, the ENGAGE trial from Biogen, that lowered amyloid, although not as much as the eMERGE trial, and didn't change clinical cognitive scores. And so the FDA thought perhaps this was an outlier. And on this basis, and I should show you also the next graph, here the new agent Bananamab, that's been, again, much in the news from Eli Lilly, an antibody directed against a pyroglutamate form of A beta, and pyroglutamated N-terminus, it also fell along this trajectory. So it wasn't just the Adukanymab data, but also the occurrence of others. And there are now two other antibodies besides Adukanymab, Bananamab from Lilly and lecanemab or BAN2401 from Eisai and Biogen that have been given fast track designation, and some decision about whether they should also be granted accelerated approval for Alzheimer's therapeutics will be made probably in 2022. I should also add that the trial that failed here, if you examine post hoc, the patients who got the highest dose of Adukanymab during the ENGAGE study, which is study 301, those are the highest dose here in dark green, had a benefit on clinical scale. Lower is better. And similar to what one saw with the high dose of the successful eMERGE trial. And people have used this as an argument that actually had more patients gotten just the higher dose in the ENGAGE study, one might have achieved a clinical benefit. But again, this has been considerably criticized for being a post hoc analysis and not being certain. And finally, I'm going to say one more thing about the Alzheimer's process. And that is that we and others have been working towards a sensitive and specific plasma biomarker that could predict cognitive decline in normal elderly. If such therapies come on the market, and they have indeed, Adukanymab is beginning to be used. Its controversy has largely restricted its widespread use so far. Then one needs simple biomarkers to indicate which patients would qualify for this treatment. It certainly isn't appropriate for other than very mild to mild Alzheimer's patients. And we've developed an assay, this is the first publication of this by my colleague Dominic Walsh, for a fragment of tau that we discovered in spinal fluid and plasma called NT1, N-terminal fragment 1. And it can be done as a capture and detector sandwich ELISA. And one can see that compared to control populations, there's an elevation of the NT1 in CSF and in plasma. This is the plotting of that for area under the curve. And this would be the ideal over here on the left. This is just a hypothetical example. And this is the actual data that one got in NCI, normal cognition, versus Alzheimer's MCI form and versus frank Alzheimer's disease. And so the area under the curve is about 0.956, a fairly significant relationship between sensitivity and specificity of this plasma tau fragment assay. We then worked with Jas Chatwal, Aaron Schultz, and others in Lisa Sperling's group at the Brigham and the MGH, and obtained these plots in a much larger number of people, 266, that in amyloid positive individuals, a high NT1 level at baseline, a single plasma baseline NT1 tau level, would predict not only the advent of cognitive decline, but the slope of that decline, much less so in non-amyloid PET positive subjects. And these various subsets of cognitive function, logical memory, the famous MMSE tests, somatic fluency, symbol coding, all of those were worse over time if a patient's NT1 level in plasma was higher. Moreover, actual structural injury to the brain, volumetric loss, shown by volumetric MRI, also correlated in the yellow dark line with NT1 level at baseline. So we believe that elevated tau in CSF presumably, but here in plasma, which is much more accessible, predicts the development of cognitive decline and actual volumetric loss. So with that, I'm going to turn to the second set of data on a somewhat distinct, but perhaps generally analogous study for the agent or the protein that we think is similar to A-beta's role in Alzheimer's disease, alpha-synuclein. So here, if we turn to Parkinson's disease, there's a substantia nigra neuron with melanin granules and this classical target image of a eosinophilic Lewy body. It's been used for a long time as a pathological correlate of the disorder of Parkinson's disease. Lewy bodies occur in other disorders as well, dementia with Lewy bodies, multiple system atrophy, and even in Alzheimer's to limited extent. So we asked the question, could some of the learnings we've had about A-beta's seminal role, we think, in Alzheimer's pathogenesis relate to synuclein's role in Parkinson pathogenesis? So if you follow this field, you know that since alpha-synuclein was cloned in Richard Scheller's lab at Stanford years ago, it was viewed as a natively unfolded monomer. That is, it did not have native structure in aqueous solution. And that, of course, is true. And it was assumed that in the cell, that would also be the case, that it was natively unfolded. And I have no of this unfolded monomer into aggregates. And these aggregates eventually form Lewy bodies in nigral neurons, and not just there, but in cortical neurons and elsewhere in the brain. Then in 2011, we came up with evidence for a more complex model that the unfolded monomer could bind to membranes and fold into an alpha helical monomer, and then that could assemble into an alpha helical rich tetramer. So this work, published in 2011 in Nature by Bartles in my lab, Tim Bartles, who's now at UCL in London, was quite controversial because it had been assumed that synuclein is an unfolded monomer. But we thought that there's still the problem of aggregation that could still come from the unfolded monomer, but there was also an alternate pathway in which the monomer could become folded on the membrane and form a tetramer that could then come off the membrane and float in the cytoplasm. So that was the theory, and I'm going to show you evidence for that. The tetramer had not been seen before in biology, and there's a reason why it had been missed. First, Tim Bartles at the time performed circular dichromosome studies on erythrocyte alpha-synuclein because red cells are very rich in alpha-synuclein. They rival neurons in the amount, and found that there was an alpha helical structure without the necessity to actually add small unilamellar vesicles of phosphatidylcholine, phosphatidylserine. One could certainly replicate Julia George's observations in the 1990s that joining synuclein to vesicles allowed alpha helical conformation. It was no longer natively unfolded. That was in vitro. We showed that synuclein isolated under non-denaturing conditions from cells, both red cells and then later brain, had an alpha helical structure to it. It wasn't just natively unfolded. Furthermore, these apparent alpha helical tetramers, shown here in blue, did not undergo time-dependent thioflavin T fluorescence acquisition in an aggregation assay. In contrast to, in red, recombinant natively unfolded monomer. So the natively unfolded monomer would aggregate, but the isolated erythrocyte tetramer put in vitro with thioflavin T did not acquire a beta sheet structure and an aggregation indicated by excess fluorescence. And so we somehow felt that alpha helical material was resistant to aggregation. Now, the notion of it being a tetramer, it came about in large part by crosslinking studies that Tim Bartles began, and then Ulf Detmer, who joined me from Christian Haas's lab in 2012 or so, began these very interesting experiments. And what he did was to apply a cell-penetrant crosslinker, DSG, that has a specific 7.7 angstrom spacing. And if there are lysines available for covalent crosslinking that have that particular spacing, one can trap molecules. Obviously, non-specifically, any molecule that has that spacing and has lysines available for DSG crosslinking. And what he found, curiously, was that if he applied the DSG crosslinker to a lysate of a cell and probed for synuclein, only a monomer was visible on Western blot, just as for 30 years, the field had assumed in biochemical experiments that what you find in a brain extract or in a cellular extract is a 14 kilodalton monomer, like Parkin is a monomer. But if you first applied the DSG crosslinker to intact cells, still living cells, you could trap a 60 kilodalton species that we showed by other techniques was tetrameric. We used sedimentation equilibrium analysis on a Beckman analytical centrifuge to show that it is 60 kilodalton, which was just the size of 4 times 14,400 Dalton monomers. And there were faint 80 and 100 kilodalton bands also on the Western blot. So intact cells, application of a lysine-directed crosslinker would trap synuclein in a tetrameric state. And notice that there's very little in between. That is, we sometimes see dimers under ideal experimental conditions when the cells are healthy. We do not see much dimer, suggesting that tetramer is a favored form. What does that have to do with Parkinson's? what Tetramer and colleagues showed was that if you look at all of the missense mutations that by that time had been identified in families with aggressive Parkinson's disease, like A15T, the first one identified by Polymeropoulos in 1997, they subtly but definitely lowered the tetramer monomer ratio. So by quantifying Western blots of this sort, you could see whether you use DSG or an alternate lysine-directed crosslinker DSP, there was a decrease in tetramer to monomer, 60-to-14 ratio, compared to the wild type situation. And that was done iteratively. It took a great many experiments to show the difference between these missense mutations causing Parkinson's and the wild type synuclein molecule in cells. We then designed a venous YFP complementation paradigm where two halves of the YFP molecule were separately labeled to alpha-synuclein, or commonly joined to alpha-synuclein, alpha-synuclein for the N-terminal part of GFP and alpha-synuclein for the C-terminal part. And these were expressed in cells together. Only when the two halves of YFP would bind together would you get a fluorescence signal for the intact YFP molecule, and that would indicate that you had at least a dimer to form. And this method was applied to synuclein, either wild-type synuclein or a series of mutations. Here in red, the E-to-K mutation at position 46 that causes aggressive Parkinson's disease, or analogous mutations in the two adjacent Kdke EGV repeat motifs that have been long known to be a feature of synuclein structure. Here in the wild type, you see them underlined. Some of them are imperfect, but in the N-terminal region, there are about 5 or 6 Kdke EGV motifs. So if you make an Kdke E to K, Kdke KGV mutation, you are on the way to Parkinson's disease. And we've done 1, 2, or 3 of these E to K mutations. Only the one in red is the clinical mutation. When these were expressed in neural cells, there was a steady K dosing related loss of the 60 kilodalton tetramer and somewhat more monomer, seen here, seen again in this example. And here, arginines were substituted for that set of lysines, since DSG is lysine directed. And again, regardless of lysine or arginine substitutions, the amount of the 60 kilodalton tetramer trapped by DSG crosslinking went down. And that's shown here by the fluorescence complementation method. And there, all the fields have, by bright field, lots of cells in them, but the fluorescent image of YFP in these cells is abrogated as you introduce these K mutations. So exaggerating the E46K mutation prevented cross-linking properly and prevented YFP complementation in intact neural cells. This actually was associated with toxicity. And here you see, for example, live cell imaging or adenylate kinase release or PARP cleavage. In all cases, these assays were associated with greater toxicity if you abrogated the ability to form these tetramers. So we assumed that tetramer formation was physiologic. We found it under wild type circumstances, and when the mutants interfered with that, that was negative. Wolf also found strikingly that when you interfered with the tetramerization, you got round cytoplasmic inclusions that were synuclein rich. And by electron microscopy, they had many small vesicles associated with synuclein monomers, almost reminiscent of a form fruste of a Lewy body in these neural cells in culture. And the amount of inclusions you got in the cytoplasm and in the neurites was dependent on how much you abrogated the tetramer. With the clinical mutation, or that plus an adjacent E to K, or all 3 adjacent regions having E to K, that elevated the inclusions. So this then came to the important question of generating an alpha-synuclein tetramer abrogating mouse to support this idea of tetramer importance in vivo. And Selkoe, who had joined my lab at the time, had experience in Parkinson mouse transgenesis and developed a 3K mouse model, first a wild-type mouse, 2K and 3K. You can see that the amount of synuclein immunostaining in the whole sections of the brain were very similar. And then microscopically in the striatum, in the substantia nigra, the 2K and the 3K, that is more tetramer abrogation by these Kdke mutations, would begin to lead to the accumulation of synuclein, both regular synuclein and phosphosynuclein, shown here at the bottom in the nigra, with round inclusions in the nigral neurons. Again, at least generally reminiscent of a possible Lewy body. And that happened in a proteinase K sensitive manner. So proteinase K treatment of the sections would still leave some residual synuclein reactivity in the 3K mouse, 2K mouse, but not in the wild type mouse. So there was a neuropathology associated in the living mouse by expressing just slightly supraphysiologic levels of the 2K and 3K synuclein mutations. But what was also striking is that, as expected, applying the DSG crosslinking to fresh brain bits from the mouse cortex showed that there was a decline in the 60 kilodalton tetramer and a gain in the monomer that could be expressed as a tetramer monomer ratio synuclein going down as a result of this mutation in the 3K mouse brain in black. And this was associated with decreased striatal TH positive fibers. So those are tyrosine hydroxylase positive fibers for the dopaminergic fibers that come from the nigra, and with an actual loss by HPLC analysis of dopamine levels in the brain. So non-transgenic wild type and 3K mouse brain shows a dopaminergic deficiency. And we found strikingly, as you'll see in this video, that there was an actual motor abnormality as a result of interfering with the synuclein tetramer formation. So you can probably guess which mouse is wild type human synuclein and which mouse is 3K mutant synuclein, namely the one on the left. That animal has a unstable gait at 12 months and has a tremor, actually a resting tremor that we can see with a titubation of the head. Now, this was quantified, and a number of different motor behaviors like clasping, wire hanging, and pole climbing were quantified. And I'll call your attention to the right side of these graphs. What we found was that the 3K animal, given saline injections, had a deficit of wire hanging compared to normal mice, and had a longer time to climb down a pole. But if you treated the animal with L-DOPA, the canonical treatment for Parkinson's motor symptoms, you could transiently reverse this. So L-DOPA treatment would partially recover the ability for the animal to hang on a wire, and it would almost completely reverse the increased time it took for the mouse to climb up and down a pole. So this was an L-DOPA responsive motor syndrome, at least in part, and the resting tremor also was less. Now, separate from this work was very interesting work done by Selkoe's lab, the former trainee of the Dawsons at Johns Hopkins, in which he examined the issue of these synuclein tetramers in an important form of clinical Parkinson's, namely glucose ribosydase A deficiency, which he found negatively affects the ability of neurons to physiologically generate alpha-synuclein tetramers. So this was quite striking, coming out in 2018. That showed that another form of Parkinson's, not just synuclein mutations themselves, but wild-type synuclein in a human that had a GBA deficiency, there was a decreased physiological tetramer formation in iPSC-derived neurons, and we confirmed that in our own work. Then we went on to do unbiased screens for what could restore the synuclein tetramer monomer ratio and what we believed were downstream effects. And in work that Serrana Fanning led, originally in a collaboration with the late Sue Lindquist before her passing a few years ago, and then Serrana came to my lab. This work with Ulf Detmer led to a lipidomic analysis that identified an enzyme that was important in regulating alpha-synuclein tetraformation, namely steryl-CoA desaturase, the rate-limiting enzyme to take stearic acid to oleic acid, so 18-0 to 18-1. This came from an extensive lipidomic analysis where fatty acid saturation seemed to regulate the ability of neurons to form proper tetramers. And a second paper just after this by Ulf Detmer's group showed similarly that steryl-CoA desaturase would alter this. So we went ahead with this in vitro lipidomic work on cultured cells to ask if a steryl-CoA desaturase inhibitor would be beneficial to our motor phenotype in these 3K, E46K exaggerated mouse model. And we found that both in wild type and in E46K cells, there was a increase in tetramer monomer ratio, even in the wild type circumstance, using an SCD inhibitor, a K compound. And that would have an effect to not only increase tetramers, but decrease phosphosynuclein accumulation, which is a classical marker of Lewy body formation in human Parkinson's disease. And so we began a trial, and this is the last data that I'm going to show you, of a Pfizer-created compound that we nicknamed 5B that inhibits SCD. So it's a steryl-CoA desaturase inhibitor, therefore lowering monounsaturated fatty acids, increasing saturated fatty acids. We showed that it was brain penetrant and that it could revert the pole climbing deficit and the number of falls from the pole and the length of endurance and wire retention. So things went in a positive direction, got better when you treated for 4 months SCD with an SCD inhibitor, these animals. And I'll show you in my last slide the effect of this in vivo treatment with oral SCD inhibitor. Again, 2 mice, one treated with placebo and one treated with Pfizer's 5B SCD inhibitor. Again, these are the 3K mice that have Parkinson effects. So the mouse on the left climbs down. That mouse was treated with the Pfizer SCD inhibitor. The mouse on the right received placebo. And after 4 months, it has great difficulty even turning around on the pole, something mice, as you saw on the left, do well, and then climbing down. And as it tries to climb down, eventually it has the risk of just slipping and falling off the pole. So this work suggests that a clinically available brain penetrant inhibitor of SCD might be something that could help people with Parkinson's. And the work has gone forward into a clinical trial, not done by us, but done by our collaborators at this company, Humanity, founded originally by Sue Lindquist, and that's now underway in phase 1B. So with that, I'll tell you that the many slides that I showed you included work on the left on human A-beta oligomers altering neurons, and that was work done by Ming Jin, Xiaomin Li, Ganesh Shankar, and several others in the lab with collaboration from Dominic Walsh and Tracy Young-Pierce. And on the right, the story I told you about alpha-synuclein tetramer monomer equilibrium being important as a Parkinson approach. Started with Tim Bartels, carried on by Ulf Detmer, and then Silke Nuber and Serrana Fanning. Let me stop there and thank you.
Thank you very much, Dr. Selkoe, for the great presentations. The questions are going to be coming up in Q&A. I already see 3 questions posted there. Let me begin with one question. In the APP cleavage and A beta production. This is happening in the membrane. Is there any specific mechanism that drives the peptide to become soluble from membrane?
Right. So you're talking about, say it again one more time.
The A beta production in the beginning, we showed the gamma secretaries cleaves
Yes.
ABP and then A beta is produced. Is there any specific mechanism known to transport the A beta peptide, which is in the membrane environment, to the solution phase?
Yes, very good question. So what we think happens is the actual proteolytic cleavage event liberates the A beta. It has the A beta, of course, is partly in the transmembrane region of APP. And depending on where you clip the APP transmembrane region, you release the A beta peptide as a soluble product into the lumen of a vesicle or the extracellular fluid. And what dictates that, Rams, is the length of the A beta. So if you cut at A beta residue 43, 44, 45, 46, there's considerable retention of the A beta species in the membrane. And we know those cleavages occur because Yasuhara in Japan has shown this remarkable processive cleavage event with the first presenilin cleavage of APP occurring at the so-called epsilon position at residue 50 or 51. And then every 3 amino acids, presenilin cleaves APP in the membrane to a shorter and shorter and shorter species. So you go from 49 to 46, to 43, and to 40. Or in another register, you go from 48 to 45 to 42 to 38. And so trimming a carboxypeptidase function of presenilin as a aspartyl protease is what actually releases A beta in the membrane. And normally, you and I release A beta in considerable quantities cut at A beta 40. That form, it does not have a long enough transmembrane domain to be retained in the lipid bilayer, and it gets released. 42, a little bit less, and we all make about 80% or 90% of our A beta that's secreted is 40, and 10% is 42.
Thank you. Hank, go ahead.
Rams, yes, I'll take the first question that's listed in the chat space. We'll stay in the amyloid area. Firstly, thank you for this beautiful talk, Dr. Selkoe, really appreciate it. This is from Rob Tycho. Are you able to produce the same effects on rat learning, avoidance behavior, with amyloid beta oligomers that are generated in vitro? Or is it necessary to use saline extracts from AD brain tissue? If in vitro oligomers do not work, why not? They
do work, absolutely. And that actually had been shown prior to our doing the experiments that I showed you. So we wanted to go from synthetic A beta to what we felt was a more pathophysiologically relevant endogenous source and used Alzheimer's brain extracts. And by and large, we ourselves have done little work on synthetic A beta assemblies. But prior to our work, and we did some work with this, we could show that synthetic assemblies, sometimes using the term ADLs, Alzheimer's related diffusible ligands that Bill Klein and many others have shown are very important. These synthetic assemblies would impair behavior of a learned behavior and would impair LTP. And actually, we also used culture medium that had A beta oligomers in it before we turned to human brain material. So the human brain material is not unique. What is remarkable about it, we published this, is that it's much more potent in interfering with synaptic mechanisms and inducing tau phosphorylation and tau neuritic dystrophy. So in the PNAS 2011 paper, that Jin et al., we showed that synthetic A beta applied identically was about 1/100th as potent. So put it the other way, the soluble oligomers from human brain were about 100-fold more potent for reasons that we do not understand precisely, but we assume that synthetic assemblies populate a number of different aggregates, only some of which have optimal bioactivity or neuritotoxic activity.
Thank you.
So I'll take the second question. It's from Rohan De Silva. So great talk, first of all. So the question is, A beta triggers the AD processes and tau progresses it. What are your thoughts on the prospects of anti-tau immunotherapy?
They are very sanguine. That is, I do believe that anti-tau therapy is going to be a very important thing to combine. And one of the interesting things about what's much in the news now, the controversy over Adukanymab as the first putatively disease-modifying agent is that it offers the opportunity to combine anti-tau with anti-A beta. Anti-tau trials have been underway for a few years. Some of them have not succeeded. Others look like they're beginning to show some tantalizing evidence of biomarker alterations in Alzheimer's patients. And so my colleagues who are in the clinical trial arena are very anxious to combine A beta antibodies like Adukanymab. Now there is an approved antibody with experimental tau antibody. And this combination approach could be a 2x2 factorial design that my colleague Risa Sperling and her colleagues are very much pushing forward now. We don't think that Alzheimer's can be treated with anti-amyloid agents by itself unless you gave them 20 years before you forgot where you parked your car.
So just to follow up on this question, so if this is the case, when do you think, at what stage of the disease it becomes A beta independent? Or if not, what factors drives the others like tau, inflammation?
Right.
So, or it's in some people stays A beta dependent, in others it doesn't. So your thought?
My view is that as much as we know right now, many forms of Alzheimer's disease begin with A beta accumulation in the brain with alkalinization. So ApoE4 is by far the most common. ApoE4 alleles might be present in 50 or 70% of Alzheimer's cases worldwide. And ApoE4 changes A beta clearance. It decreases it and allows more aggregation in the extracellular fluid. So my view of things is that A beta is the initiator years before the first symptoms, and then there is tau alteration. And that tau alteration is what's much more proximal to the dementia and is much more, quote, important for the actual clinical impairment. Without tangles, I don't think A beta effects very much injury to neurons. It works by increasing tangle formation and tau alteration. And so at least at this point of view, perspective, A beta starts decades before clinical symptoms. Eventually tau accumulates, achieves a certain maximum. Everything is quantitative, everything is a continuum. And if you get enough injury, you begin to see clinical effects. And treatment would best be done 15 or 20 years before you and I get memory failure. How would we know that? We would know it from our family histories. Let's say we have a mother or father at Alzheimer's. We would know it from genetic analyses. Obviously, the rare autosomal dominant mutations in presenilin and APP can be picked up at birth if one wanted to, and one could do it by ApoE4 genotyping. And there are, of course, trials underway in people who are pre-symptomatic, who do not yet have Alzheimer's symptoms. But I think that once you have mild symptoms, A beta lowering by itself may be too little too late. And there you'd want to also have tau lowering, and you'd want to have a microglial alteration or an astrocyte mediator that would decrease that problem. So it ultimately will be a multi-therapeutic approach, but I would have to say right now, the best evidence in my mind is very early A beta lowering, arguably a decade or more before symptoms. And that can be determined, for example, with the blood test that I mentioned briefly.
Hank, I know you got to leave, but would you like to ask your question before you leave?
Sorry, I was muted.
Go ahead, ask your question, Hank.
You want me to ask a question? Okay, I will do so. Let's go to synuclein. Let's see if we can find a synuclein question here for you. There were a couple that were related to synuclein. I will start off by saying, firstly, Dennis, the cross-linking shows beautifully that you can capture the tetramers in those cells where you have these K mutations. Presumably there are higher order aggregate forms that are also captured by cross-linking, but we're not seeing them in your gels. So are they not captured? They're not coming into the gel? What's happening there?
Yeah, it's a great question, Hank. And what we think is that the DSG cross-linking of 7.7 angstrom doesn't trap very efficiently abnormal aggregates. There should be tetramers that are abnormal tetramers, whereas my work has been emphasizing physiological tetramers. So we've looked, when we aggregate tetramers, Kdke mutations, we know that there's aggregation going on. The mouse has Lewy-like structures in its neurons, and it has a dopaminergic deficiency, et cetera. But we are not able to use DSG cross-linking to trap those others. I don't doubt that other cross-linking agents might trap those, and we can see them as in SDS gels as gel-excluded material, as you would expect from an amyloidogenic material. But they're not trapped by the DSG cross-linking. And we've often been asked, we don't get it. What about pathological oligomers? They must somehow be conformed differently than this. And this is all operational. We looked at many cross-linkers, and DSP and DSG have the spacing that traps the physiological, but not the pathological oligomers, which is not
Thank
to
you,
say
Ram.
that
Since I need to go, I'll just ask the question
Yeah.
from Mauricio Torres, which is great talk. Can the consumption of saturated fatty acids change the formation of alpha-synuclein tetramers in the mouse Parkinson models?
Yeah, we're testing that now. We think that a dietary change in saturated fatty acids would be beneficial within reason. There are other negative effects for cardiovascular health, but a increase in saturated and decrease in monounsaturated fatty acids in the diet could potentially be a treatment. And we are embarking on that. Those are difficult studies to do to make sure that we have good nutritional balance and we're not distorting things too much. Certainly the SCD inhibitors, which have now gone to the clinic, look like they can do it pharmacologically, but we prefer to do it in a more physiological way with diet.
Thank you, Hank. Woody, would you like to ask your question or read a question? Woody? Gazette, yeah, go ahead.
Yeah, I'm here again. Yeah, actually, this may be a follow-up on what was just asked about nutrition. What is your opinion on the effect of lifestyle polyphenols, any amyloid inhibiting compounds in terms of nutrition? How are you convinced in the ability to modulate the aggregation of A-beta as a lifestyle effect on the disease?
It's a great question. The truth of the matter is, for all the time that I've studied Alzheimer's and I've followed hundreds of patients with the disease also, I've not been able to find a lifestyle factor in everyday life on this planet that clearly protects people from Alzheimer's disease or promotes their healing. I don't doubt that there are factors such as the Mediterranean diet and healthy non-Western diets that can be beneficial, that seem to be associated with less likelihood of getting Parkinson's, actually Parkinson's also, but also Alzheimer's. But that may not be directly on the amyloid process. There may be some beneficial effects of healthy nutritional diets like Mediterranean diets for neural health in general, and also to prevent from cardiovascular disease to the brain, cerebrovascular disease. And so since so many patients who are 75, 80, 85 with Alzheimer's also have microinfarcts or vascular abnormalities, things in the nutritional sphere that help that would give them less total injury to the brain. But I have to say that actual food substances or nutritional supplements that really make a difference in the progression of Alzheimer's, in all the years I've studied patients with this, I've not seen good evidence of that. In the case of the Parkinson question that I answered earlier, it could be that pushing towards saturated fatty acids a little bit would be beneficial, but we don't know that.
Is there any epidemiological evidence for this, or for the fatty acid, or you are just studying it?
No, there's no epidemiological evidence in Parkinson's for the fatty acid, to my knowledge. In terms of Alzheimer's, there have been claims that certain types of diets or certain, curcumin, for example, in Indian foods could be beneficial. And when you directly test that in experimental controlled trials, it's been difficult to show that a food substance really slows down or prevents Alzheimer's disease. With the fatty acid situation in Parkinson's, I have no evidence that that's ever appeared. I'd be interested in knowing if someone else knows.
If I can have another question, Ram?
Sure.
A more general top question. As all know, you are one of the really founding fathers of the amyloid hypothesis. And beta amyloid is only one example of many proteins and polypeptides that tend to form these amyloid structure.
Right.
On the other end, your data on LTP, LTD, the glutamate reuptake is very convincing. So where do you think there's this specific role of, let's say, A-beta as islet amyloid polypeptide, other amyloid forming peptide in terms of amyloid architecture versus specific activity of, let's say, A-beta?
Yeah.
Where it's the generic amyloid phenomena and where it's the it's the specific A-beta effect.
Yeah, so Bruce Yankner, maybe 15, 20 years ago, did some very interesting reductionist experiments applying to neurons either A-beta synthetic aggregates or islet amyloid polypeptide or amylin aggregates, and found that the neurotoxicity was almost the same in an in vitro reductionist system, that you couldn't tell hardly any difference, that when you had excess beta sheet rich fibrils applied to cells directly, the cells weren't happy, regardless of what the primary structure of the amyloid was. If you think of transthyretin amyloidosis, which is a fantastic thing, there are 3 FDA-approved drugs for transthyretin amyloidosis, which happens in the heart and the peripheral nervous system. And there you interfere with a transthyretin misfolding into beta sheet rich structures, and you really help patients' neuropathy or cardiomyopathy. And there's a very different structure. I think that amyloids are rather promiscuous, and that a number of different protein structures can injure cells when they're present at high levels and they bind to the membrane, particularly if the fibrillogenesis process happens while the monomer or dimer is first bound to the membrane, intercalate in the membrane, and then you add more monomers or dimers, you get an aggregation that's actually damaging the physical structure of the membrane when that happens. So in short, I don't think the observations we made are unique to Alzheimer's. The reason that A-beta is the bad guy, I think, in Alzheimer's is because of beta secretase. That is largely restricted in high expression to neurons. There's a little bit of expression in the pancreas and other tissues, but by and large, it's a neural. And so when you ask me, why is this A-beta problem could be happening all over the body, APP is expressed in all cells, why is it in the brain? Probably because beta secretase is very efficient as an aspartyl protease to generate the first cleavage, followed by the gamma secretase, which again is ubiquitous, and the A-beta accumulates in the brain. It accumulates a little bit in other tissues. We showed this many years ago, but very little, and the process doesn't go forward. So this issue of the relationship between a certain amyloidogenic protein and the clinical syndrome it induces is very elusive. It's very hard to say. And I think that if you engineered an animal to make a lot of amylin in the brain, you'd get an Alzheimer-like phenotype with synaptic plasticity abnormalities.
This is exactly what I wanted to ask you. Okay, maybe we'll move to some of the questions in the Q&A. There's one from Bin Shu. Thank you for a very interesting talk. Can you comment on your plasma-based NT1 tau biomarker assay versus p-tau 181 and p-tau 17 blood-based Alzheimer's disease test reported recently, 2020 in AD field? Do the new assays have similar or better specificity and or sensitivity?
So we're comparing head-to-head the NT1 fragment of tau that I highlighted in our work to the PT217, the phosphothreonine 217 of tau, the phosphothreonine 181. And we don't yet know which one would have the greater sensitivity and specificity. We know the field of Alzheimer's is very excited about the phospho-tau 217, but my colleague Lei Lu, who did a lot of the biomarker work I discussed, is showing that it looks like the NT1 assay might be at least as good as a phospho-tau 217, and that also the 217 is not necessarily the optimal phosphoepitope to detect in plasma. Again, we have to distinguish what we can detect in spinal fluid, which is more straightforward, compared to plasma, which is a much more complex matrix. And yet, we don't want our patients to have lumbar punctures routinely. They can't do that. They won't do that. So we have to rely on plasma. And in the plasma, the answer to the questioner's point is not settled as to which assay is. And then I should make life more complicated by telling you that just 2 days ago, we published a paper on detecting oligomeric A beta in plasma. So that's sort of cool, we think. You can have a selective sandwich ELISA that quantifies the oligomers and doesn't see monomers. The capture antibody is unable to capture monomers successfully. I mean, it does at several hundredfold higher amounts. And so that oligomer level could be very interesting in the plasma 2 decades before patients develop symptoms. These tests are all in competition now, and they have to just go through a very laborious process of head-to-head comparisons to see which ones. And it couldn't happen faster because it looks like Lilly and Eisai are pushing ahead with the FDA to try to get their accelerated approval a la Adukanymab.
Okay, let me ask a question from the Q&A. This is from Christian Grisinger. He has 2 questions. I think one of them answered already. The second one is, you showed BAN2401 with Adukanymab. Adukanymab, according to my knowledge, caused brain edema in quite a number of patients treated. Did this problem occur also with BAN2401?
So the question is, what with BAN2401 does what Adukanymab did in mice? In mice or?
In patients.
In patients. So lecanemab or BAN2401 looks quite good and probably at this moment better than Adukanymab. Of course, there are different data sets, different patients. It's not a head-to-head comparison, but it looks like lecanemab BAN2401 is at least as good, if not better, than Adukanymab in terms of clearing amyloid. Clearing amyloid, they probably both do similarly, but in more clinical benefit. And very importantly, it turns out BAN2401 has much less of the principal side effect of antibody treatment, something called ARIA-E, amyloid-related imaging abnormality that I didn't talk about today. But it has about 1/3 the level of abnormality or adverse effect like lecanemab does than Adukanymab. So we need to have the 2nd and 3rd and 4th antibody treatment approved so we can compare them in the clinic and see, you know, in MS, multiple sclerosis, they started with 1 in the 1990s and now they have 15 FDA approved agents. So I think that lecanemab looks right now, let's put it this way, if I had this disease, I'd probably want to be on lecanemab just as well as Adukanymab.
Thank you. Rakesh, go ahead.
Yeah, so the next question from anonymous attendee, very nice talk. So thank you for good talk. The question is about the time alpha-synuclein aggregates are maintained in the M17D cells. Or in other words, what is the effect of the alpha-synuclein overexpression on M17D cells viability? So,
yes.
Can you comment?
Yeah, so the M17D cell model that I spoke about does show neurotoxicity. I mean, these are dopaminergic human neuroblastoma cells, so they're not primary neurons. We've done a lot of this work in iPSC-derived human neurons. And so you see toxic effects on the neuron if you abrogate the tetramers and you accumulate monomers. Now, people have argued, well, maybe it's got nothing to do with the tetramers. Maybe it's true, and unrelated. The tetramers go down, fair enough, but there's other reasons why these mutations you study are causing trouble. There's increased membrane association of the synuclein. And I agree with that we can't be sure that the tetramer abrogation is the key feature, but we use other mutations like G51D that causes a nasty form of Parkinson's. It doesn't have increased membrane association. It also lowers tetramers and it causes neurotoxicity in M17D and other cells. The most important thing is not to rely on M17D cells, but go to human neurons. And they have 4 copies of the wild type synuclein gene rather than the normal 2. And they get a very aggressive form of Parkinson's, this family from Iowa that's been known for a decade. And we've obtained iPSCs from those patients and cultured them into neurons. And they have all the abnormalities I talked about in the M17D, but just more subtle. They get abnormal, they can get inclusions, they can certainly get abnormal phosphorylation of synuclein, decreased tetramer monomer ratio. This is just a matter of too much wild type synuclein without a missense mutation. And so that solves some of the critique that we've rightly gotten. This is a weird mutant you have there with these 3 Ks. Who knows what that's doing? We can show very similar pathological effects from wild type synuclein in human neurons taken from synuclein triplication carriers.
So just one quick question. Do you know if beta and gamma synuclein also form tetramers as functional? I'm not sure. I read it somewhere.
They do. We published that in JBC in 2013. So when Detmer first described the crosslinking method, he did this to beta and gamma synuclein, tested that, and they have beautiful tetramers just like this. So we don't know why it is that beta synuclein isn't causing a form of Parkinson's disease. But the 2 homologs, they have these E to K, they have these Kdke EGV repeats, and they also form tetramers.
Woody, your turn.
You're muted.
Woody, unmute yourself.
Maybe once again, because I was brave enough to ask about lifestyle, there's a question about exercise. Is there no clear evidence that it's helpful? I assume that your answer will be no, but I will let you answer.
Yeah, there is evidence that exercise, both physical and mental, is probably helpful in staving off late life cognitive decline. So definitely physiological exercise, aerobic exercise is associated with less dementia overall. There's also some evidence that social interactions, complex, meetings of the sort we're having today, where you put your neurons around topics you don't really think about often, those kinds of things are beneficial for staving off the likelihood of dementia. They're probably not going to work if you have a presenilin mutation, or if you carry 2 ApoE4 alleles. That's not going to be enough. But it could well be for conventional late life dementia, if there is such a thing, that exercise, both physical and cognitive, could be helping the situation. And Dale Bredesen has wrote a book, The End of Alzheimer's, touting these experiences and exercises. I don't think they'll keep you from getting the disease if you have a family history of it. But maybe if it's a very mild predilection, exercise could be helpful. So there's not a doctor in the world who wouldn't say that exercise is helpful.
But do you think, what do you think is the mechanism? You think it's really the formation of new neural connections, or some people, they offer you to do things that your brain is not used to do. Let's say walking backwards, or brushing your teeth with not with your
left
dominant hand.
So
I'm not sure if there's real data about this, or it's only.
Well, there is evidence of neural networks being increased by exercises, by certain kinds of activities. There is the evidence clinically that people who have strokes, if you tie their good hand behind their back, they have to use their bad hand, and they get more and more evidence. And there's electrophysiological evidence that they're increasing connectivity from the opposite side of the brain that's been damaged. So I think there's quite a lot of evidence that neuroplasticity is powerful. And then we have the notion of physiologic reserve. For example, we know that there are patients who have a lot of amyloid in the brain and are not having trouble thinking. They have lots and lots of amyloid plaques, equivalent to the people who don't have, equivalent to people who have Alzheimer's, but they don't have cognitive decline. And one beautiful study by David Brody at that time at Washington University showed that the oligomer amount in the plaque is less in those people who do better, who do all right. The oligomer level is per plaque is low compared to the people who have clinical dementia. So there is this notion that you can get the pathology, and some people handle it well. And almost certainly learned activities, exercise, strengthens your neural networks and makes you relatively more resistant. Again, I don't think that's a cure for Alzheimer's, but it could very much help lessen the burden of dementia.
Let me take this question from Q&A from Dr. Carmelo La Rosa from Catania. The tetramer of alpha-synuclein is not toxic, while the monomer is toxic. The tetramer is in equilibrium with the monomer. Do you have a hypothesis of what factors can induce the dissociation of the tetramer to monomers?
Yeah, it's a wonderful question. So I really appreciate your very sophisticated audience paying attention to all these experimental details. We think that one of the things that really controls the tetramer to monomer equilibrium in normal healthy neurons is fatty acid levels. And so the levels of monounsaturated fatty acids like oleic acid, palmitoleic acid, almost certainly are something that if they're too high, you go away from tetramers that are healthy and you get more free monomers. And then of course the free monomers can themselves aggregate over time into other kinds of oligomers that are toxic. There could be a toxic tetramer, which I distinguish from the tetramers we're speaking of today, which are physiologic. So I'd say lipid alterations, and from in that vein, it's very important to cite the work from Basel and Amsterdam, some beautiful work on the notion that Lewy bodies in Parkinson's have profound lipid membrane alterations that have been largely overlooked. So the folks on this call are very interested in protein misfolding and in amyloid diseases. And Parkinson's is an amyloid type disease in part, but it's also a very profound lipid membrane alteration. And you can see fragments of membranes that are altered and lipid profiles in the Lewy bodies in Parkinson's patient's brain. So recently, we've written a couple of review articles suggesting that is Parkinson's a proteinopathy or is it a lipidopathy or is it both? And my feeling about what influences the tetramer monomer ratio, what we know so far is levels of monounsaturated fatty acids. Clearly, we can take cells that have a Parkinson abnormality like a synuclein Kdke mutation and condition them in myristic acid 14-0 and find dramatic improvement in the cellular phenotypes just by conditioning in myristic acid. I would like to give myristic acid to our mice and see if their tremor gets better, et cetera. And we're going to do just that. So I think that the most likely thing that regulates the normal tetramer monomer equilibrium and pushes towards healthy tetramers is having less monounsaturated fatty acids, not none, but somewhat less.
So
maybe a follow-up on this. You discussed the fatty acid, but there's also quite a lot of work on cholesterol and amyloid. Right. For example, from late Chris Dobson and others, right, speaking about oligomers of alpha-synuclein and the interaction with cholesterol. What is your view about this?
I think it's also very important. And I think there's a cholesterol binding site in synuclein that Dobson and others have focused on, or had focused on. And I have no doubt that cholesterol manipulation, and if we turn to the Alzheimer's story, we know that animals that are treated with statins that have a genetic predilection to Alzheimer amyloid will get less amyloid. So, and by the way, earlier when we asked about diet and exercise, there is some evidence even in mice that exercise itself lowers amyloid protein levels or makes it less oligomeric. And synuclein, I think there may also be some evidence. So I would say that cholesterol homeostasis is important. On the other hand, clinicians have tried to use Lipitor, Zocor, and other HMG-CoA reductase inhibitors to try to treat Alzheimer's, and it hasn't been greatly beneficial. I mean, there might be a little bit of evidence. I reviewed some of that evidence in past years, and it's a close call as to whether chronic statin treatment really slows down Alzheimer's.
I assume that nowadays so many people are taking statins that
That's right.
A smart person with the knowledge of handling the data could find out
I think if
there was some correlation.
It is, and we could find that out more definitively than the studies that were done a decade ago. And I don't doubt, and I would certainly encourage my patients to take statins if there's any other reason to take them for lipoprotein purposes. It would be another benefit that might help their Alzheimer's.
So on this topic, there is another question from anonymous attendee. Do you still find evidence for colocalization of beta secretase, gamma secretase in the membrane, supporting the processing of APP? And do you feel membrane heterogeneity and so cholesterol levels play a critical role in the colocalization of these enzymes?
Yes.
And before, there is another question related, maybe I can sum it with it. What about the ABP intracellular ABP fragment?
Right.
Their role. So there's a question.
So this is a topic dear to my heart. We actually described in the Journal of Cell Biology in 2019 the existence of a beta gamma secretase complex in which you can pull down from brain a high molecular complex. It's probably greater than 5 megadaltons, approximate size by size exclusion chromatography, that has both beta secretase and gamma secretase. You can incubate these pull down fractions at 37 degrees and generate a full array of A beta peptides from this high molecular weight protein complex. I don't know what else is in there in that complex, but there certainly is active base and presenilin gamma secretase. And when you pull it down from mouse brains that have mutations or cells that have mutations in presenilin, you get the mutant profile of A beta, more A beta 42 and 43. So the two enzymes work together in a high molecular complex that makes it very efficient to pass the substrate from base cleavage to gamma cleavage. And that probably happens in other situations, like in Brown and Goldstein's pioneering work on site 1 and site 2 proteases for SREBP cleavage. Those two can also occur together, site 1 and site 2 proteases that act sequentially. And so I think the C-terminal fragment of APP, which is the last 99 amino acids of APP, many people have felt that's the toxic bad guy. But the irony is that if that were the direct cause of the Alzheimer's problem, then buildup of the C-terminal fragment would lead to less A beta, because the C-terminal fragment of APP is the substrate for gamma secretase generating A beta. So the A beta then would be beneficial because you would have less intact C-terminal fragments. I don't think that's how it works. And I don't think that the intact C-terminal fragment, by which we mean C99, is the toxic moiety. Because while it can accumulate and can be toxic if you overexpress it in the cell or an animal, you get less A beta. But the brain in Alzheimer's, by PET imaging and many other studies, accumulates more and more A beta over time.
Thank you. Shall
Woody.
we stop right now?
Woody?
Any Q&A? Is there an anonymous attendee? Is there a biomarker for early detection of A beta oligomers, similar to the tau NT1 you presented?
Right. I mentioned to an earlier question briefly that we just published a paper that folks can look at in the journal Alzheimer's and Dementia that we think is the first specific assay for detecting oligomeric A beta in plasma. We had previously published in 2015 an assay for CSF. It was not sensitive enough to detect it in plasma. The new assay is more sensitive, and it's a sandwich immunoassay that has a capture antibody that is selective for oligomers and doesn't see monomers well at all. And you can use it in plasma because you can dilute the plasma 8-fold, 16-fold, maybe 32-fold, thereby diminishing matrix effects. Plasma is a very complex analyte, obviously, and a difficult thing to study. So we've done quite a few patients with that, but only normals. We've not yet learned whether the oligomer level in the plasma is elevated in patients with Alzheimer's compared to controls, or goes up pre-symptomatically. That's all what we have to do, but the assay now exists and it can be seen. It's Liu et al., L-I-U et al., Alzheimer's and Dementia, October 2021.
There's another question from Victor Shing about how might the tetramer formation of synuclein relates to its quote unquote normal function. And I may use it for even make the question a bit broader. So as we all assume, amyloids were not created by evolution or God for us to get Alzheimer's disease.
Right.
Do you think about
Absolutely.
the general property of beta amyloid and amyloid and proteins that tend towards amyloid aggregation in general.
Right. So in the example of synuclein, molecular modeling suggests that the tetramer is an energetically favored assembly form. So the protein likes to be in a tetrameric form, and it's probably kept that way by binding a small limiting factor, such as a lipid, for example, maybe a fatty acid. And the tetramer probably is a normal functional property that is beneficial. The function of synuclein by many in the field is believed to be managing or regulating trafficking of vesicles, small vesicles, to which synuclein binds to the external surface of the vesicle. And there's one nice study by Subhijit Roy at University of California, San Diego, showing that vesicle movement regulated by synuclein only really works when you allow dimer or higher tetramer assembly. The monomer doesn't regulate the synuclein property to retard the movement of vesicles. So to the extent that synuclein seems to be an agent, a protein that regulates the kinetics of vesicle movement from the cytoplasm to the surface, to the membrane, and release transmitter, it looks like the tetramer is necessary for that function. And the monomer doesn't do that as well. He did some very nice studies with a tetramer abrogating synuclein, and it didn't work when you didn't allow tetramer, or he could only see dimer assembly. He saw by fluorescence complementation on the surface of vesicles. And I think in general, A beta, for example, could have some physiological functions as a growth factor, according to some studies. It's the excess and misfolding to too much of a filamentous or pre-filamentous form, a protofibrillar form, that seems to be beneficial when you accumulate it on the surface of cells or inside cells. Synuclein is largely intracellular, A beta is largely extracellular. So I do think that the normal function of these proteins is one topic, and they have normal functions, but the accumulation of misfolded forms is another topic, and that seems to be more associated with the disease. So in our case, synuclein, normal tetramers are physiologic. Less tetramers and more free monomers leads to aggregation in abnormal ways that seems to be detrimental to dopaminergic and other neurons.
And if I use it for a more general question, you see from time to time work that says that Alzheimer's disease is a herpes-related disease. Other work that it's a periodontal-related disease. Do you think that A beta or mechanism that lead to A beta aggregation has to do with some kind of a viral load or bacterial load? What is your opinion on this?
Right. So my personal opinion scientifically is that the evidence is not strong for an infectious pathogen. There is some evidence, no doubt. And when I debate this with my colleagues, they point out that there is evidence that if you put A beta with bacteria, you can opsonize the bacteria. The A beta can bind to it. It doesn't surprise me that hydrophobic assembly, like amyloid filaments, would somehow retard the spread of bacterial infection. But the question in the human is whether this is acting every day. The amyloid is actually beneficial in some way to retard infectious processes. That's one question. I don't think so personally. And the other question is whether herpes virus of some sort or another is something that stimulates the A beta or the tau or other inflammatory process in the brain. And since herpes simplex virus is so ubiquitous in humans, it's been difficult to get clean epidemiological evidence that Alzheimer's patients have definitely more herpes infections or more herpes carrying. So my personal opinion is that I think microglial inflammation, astrocytic inflammation, is a very important factor, and genetics supports that. There's a lot of genetic factors that predispose to Alzheimer's that are microglial surface genes. That's no question. That there's a viral or bacterial pathogen that accumulates in the brain, I think is far less likely.
There's one question in the chat box from Dr. Beena Krishnan. I'm not sure if we addressed this or not. Do we know which proteases are responsible for in vivo generation of NT1 that is released in plasma?
Yeah, that's a great question. So we don't know, and we really have to purify that fragment from CSF and plasma and then, which is no difficult, it's not an easy thing, and then sequence it, find out where the cut point is, and then decide which proteases. It might not even be a single proteases, but right now, the precise C terminus of that NT1 fragment is probably around 184, 185, 200 in the synuclein molecule, and in the tau molecule, excuse me, in the tau molecule. And I don't know what proteases cause that cleavage, but it would be very wise to know that. However, what we've only seen so far is that the NT1 fragment is a marker of the Alzheimer's process, not necessarily causative. So I'm not sure inhibiting that protease would necessarily be therapeutically beneficial. We simply don't know what enzyme, what protease generates that fragment. And that might not be pathogenically important, but it is a very nice correlate of the disease.
So I have a question on similar, the plasma versus brain. Is it an inverse relationship, or it's, if it increases in the brain, it increases in the plasma? And does this affected by the vascular health? Because there is a lot of the lymphatic involvement in the clearance of the oligomers or the amyloid aggregate. So where the vascular health fits in developing these assays?
Yeah, I think it's, again, a wonderful question. I personally believe it's very important. So vascular health and microvascular intactness, the blood-brain barrier status, these things really help regulate the processes we've been discussing, especially in Alzheimer's, which is, of course, an extracellular amyloid, like most classical amyloids are, immunoglobulin amyloids, transthyretin. It's an extracellular accumulation, and the clearance, we know, comes out of the extracellular fluid and into the vascular system. So you need efficient clearance mechanisms. It could be that the lymphatics of the brain that have been very exciting to hear about are part of the way that you flush amyloid aggregates out, especially A beta, which is largely extracellular. And then there's the overlap between vascular disease of the brain and Alzheimer's. So that's really like a Venn diagram, sort of, where there's a problem in brain health from vascular disease, there's a problem of Alzheimer's, and if you have both, you have a double whammy, and you're really much more vulnerable. So understanding how to keep microvessels healthy is very important. I don't believe the entire Alzheimer's biological process is caused directly by vascular abnormalities. I think it's largely neuronally oriented, because that's where beta secretase is, and that's where tau is, and that's where synuclein is, in the neurons. But I do think that microperfusion, clearance from the brain, very important topics. And so I think there's a vascular aspect to Alzheimer's pathogenesis that needs to continue to be studied very closely.
Thank you.
I think we should, that's fantastic Q&A session. Thank you very much, Dr. Selkoe, for your time and sharing your results.
Amazing talk.
Thank you.
Okay. Well, great to have, thank you so much for all your attention. Fabulous questions. That means people really paid attention to the details, and I'm very, I'm very gratified by that. And I could go on for another hour, but I think we've all had a long time. So I'm grateful for your invitation.
Thank you
very much.
Thank
you all for joining us, and we'll see you on October 18th. We have a mini symposium, like 10 speakers are presenting on the roles of metals on amyloid disease. So these are experts in this area. So we are going to thoroughly digest the topics in that particular area. See you all then. Bye.
Thank you. Bye-bye.